Hello, my lovely anatomist and physiologist, Michele Glass here, and we are ready to dig a little deeper into our chapter 19 of our OpenStax textbook. And so we're picking up here with what is described as the cardiac cycle. And I have this lovely diagram for us coming directly out of the OpenStax book, which I have linked in the video description. So to get started, when we're talking about cardiac cycle, what we're talking about is basically the, let's say, activity of the heart, let's say for one heartbeat. This is my definition. So we're looking at the contraction of the atria and the contraction of the ventricles. Now we're going to be using these terms systole and diastole. And so we need to become very familiar with these two terms. And they're sort of opposites of each other. So systole, you should think of this as the contraction. So this is when the cardiac muscle tissue is actually contracting. And let's say pushing blood. And then the diastole we should think of as the cardiac muscle cell is in relaxation. And here we might think of this as the chambers are filling. Okay. And so as we look at our diagram, what we're seeing is a picture of a heart, and it is a cycle. So we are going in a circle, and we're seeing differences happening in the heart at each level. So let's begin where it says start. And so notice at start, we have our, I'm going to call them the AV valves. Remember, this is the tricuspid valve on the right side of the heart and the bicuspid valve on the left side of the heart are both closed. And then notice also our semilunar valves. And these are, remember, pulmonary semilunar at the base of the pulmonary trunk and aortic semilunar at the base of the aorta. These are going to be closed as well. Notice that the heart is not having any areas highlighted. So we would consider this to be in diastole. And that's going to be true for both the atria, those are the top two chambers of the heart, and the ventricles, the bottom two chambers of the heart. Notice that you have black arrows indicating the movement of blood. So the black arrows in the heart diagrams are showing the movement of blood. So we have blood moving from the superior and inferior vena cava into the right atrium. We have blood moving from the left and right pulmonary veins into the left atrium. And then notice you have blood that's like moving down toward the heart in both the pulmonary trunk and the aorta. So it's that movement of the blood down into those larger vessels that actually close the semilunar valves. But notice that the ventricle has no activity. There's no movement of blood in or out of the ventricles, right? And the ventricle is in diastole. Diastole is the relaxation. And so let's highlight here in this early diastolic ventricular diastole, we have what's described as the isovolumetric relaxation. So let's go with that terminology. Isovolumetric relaxation, and the prefix iso means same, and so here it's saying like the ventricle has the same volume of blood in it, meaning it's not getting any extra blood entering, it's also not having any blood leaving. So this is the starting point for our cardiac cycle. where the atria and the ventricles are relaxed, but right now the ventricle has no blood that's entering. Okay, let's move next to when we get our ventricular filling. And so I'm gonna grab a different color pen and I'm gonna circle this next stage. Okay, so the heart is still in diastole, which means relaxation. But notice now that we get the black arrow showing the movement of blood moving from the atria directly into the ventricles. So what's happening here is the pressure has dropped in the ventricle such that the pressure of the atria is greater than the pressure in the ventricle. So remember, the atria have been gaining blood and the ventricle had all of the valves closed. At this point, what we see is the pressure of the atria is greater than the pressure in the ventricles, and this pushes open the AV valves. So in the ventricular filling, in this point of the diagram, we have AV valves open. And AV valves, remember, are including both the tricuspid on the right side and the bicuspid on the left. And what we see is that the blood is entering the atria. So you have blood flowing from the body through the superior inferior vena cavae into the right atrium. And then you have blood from the left and right pulmonary veins entering into the left atria and is flowing directly into the ventricles. It's flowing directly into the ventricles. And so what we see here is that this is what we can call our passive filling time. There's no contraction in the heart right now. So this is our passive filling time. And what we're going to see during this passive filling time is that 70 to 80% of the blood that ever makes its way into the ventricle is doing it here during this passive filling time. So I'm going to actually, in my ventricles, write 70 to 80% full at this point. This is super significant. So what this is telling us is that diastole period is important and getting the majority of your blood into your ventricle. That diastole, that passive filling time, is important in getting the majority of your blood into your ventricle. The next step that we have happening is the atrial systole. Systole means contraction. So notice that you have these sort of red arrows pushing down on the atria, and you get this kind of purplish color to the atria. And what that's indicating is the atria are under contraction. And so now we're pushing the rest of the blood in the atria into the ventricles. Notice that the only black arrows we have showing the movement of blood are from the atria to the ventricles. So when the atria contract, that kind of stops any further movement of blood into the atria. And when the atria are contracting, that's really pushing the remaining blood into the ventricles. So here we're going to get, it can be described as the atrial kick. I actually read that for the first time in our OpenStax book, and I really like it. This is that final 20 to 30% push of blood into the ventricle. This is that kind of final last effort push to get blood into the ventricle. Okay, and so then that takes us to the ventricular contraction. So notice in our next picture, the atria are in diastole, that means relaxation, but we get these red arrows on the outside of the ventricle and we get this purple color to the ventricle. And so what we're seeing in this next picture is that the ventricles are beginning to contract. And as those ventricles contract, as they enter systole, the first thing that we see happening are those AV valves closing. Okay, and remember, I'm using that as my kind of all-inclusive term to talk about both my tricuspid valve on the right side and my bicuspid valve on the left side. So what we see happening is as the ventricles contract and begin to push and build pressure, the first thing that happens is the blood starts to like get pushed up back towards the atria. And this pushing of the blood back up towards the atria actually closes off the AV valves. Remember, you have those muscles, the papillary muscles in the ventricles, and you have those chordae tendineae kind of like pulling down on those valves so that they don't push all the way up into the atria. And then that's called regurgitation when the blood is moving in the wrong direction. So in a healthy heart with healthy valves, we see that those functions of the papillary muscles and chordae tendineae keep those AV valves closed. Now, during this period, we have what we can call the isovolumetric contraction. And I see that my highlighter color is not really great on this green background. So I'm going to highlight the isovolumetric contraction as our term here. And so this is that same sort of idea, but this time we're talking about the ventricle under contraction. So the ventricle is contracted. Isovolumetric means same volume. So here what we're seeing is the AV valves are closed, and let's also list for ourselves that the semilunar valves are closed. So the semilunar valves are closed. And what I'm going to do, y'all, is post just this diagram for you in your video description so that you have an easy to print off version that you can label. Remember, it's coming directly from our OpenStax textbook online. So you could also pull it off of that website if you prefer. Okay. So here we are in the isovolumetric contraction where we have the valves of the ventricle are closed, meaning no blood is entering anymore. Like whatever blood we were able to get into the ventricle at this point, that's what we have. And then no blood is leaving at this point, right? So we'll actually talk about this volume of blood. We'll talk about this volume of blood as the end. I'm going to make that be a capital D. End diastolic volume, what we can call the EDV, or we can talk about this as the preload. So this is our starting point, how much blood we have in the ventricle when relaxation ends. So like at the start of contraction, how much blood do we get in the ventricle? Meaning, how much did we get in the ventricle during that passive filling time? How much blood did we get in that final atrial kick, that final push of blood? That's what we mean by the end diastolic volume, or the EDV. The next stage of our cardiac cycle is referred to as the ventricular ejection. And so notice at this point, you get enough pressure building in the ventricles from the ventricular contraction. So we still get those red arrows on the outside. We still get this purple color to our ventricles showing that we are contracting more at the ventricles and more effectively, like pushing. And what we see happening is we open the semilunar valves. So the ejection here is talking about that push of blood, pushing open the pulmonary semilunar valve and pushing out through the pulmonary trunk, pushing open the aortic semilunar valve and pushing out through the aorta. Notice that we have those black arrows showing the flow of blood out of the heart. This is our ventricular ejection. And what we'll see is the amount of blood that leaves here is called our stroke volume. So we're going to label this the amount of blood that leaves the heart. Okay. And then notice that takes us directly to where we started. So this is a circle, right? So it takes us right back to where we started. So now we're seeing that the ventricular systole stops. We're entering into ventricular diastole. So we're talking about the ventricles under relaxation. And we see that because the pressure is no longer pushing the blood out of the heart, gravity brings that blood to fall like back. So gravity in the pulmonary trunk and in the aorta, the blood like falls back towards the heart. And that's what's actually going to close off the semilunar valves. So the semilunar valves are closing because of the blood flowing back towards the heart because it's no longer being pushed out. Notice, remember, we're in the isovolumetric relaxation, right? So we have the AV valves are still closed, and we see that the semilunar valves have just now closed because the ventricles stop pushing them open. And so what we see is that the heart is a super effective pump, but it's not able to pump out every single drippy drop of blood. The heart cannot pump out every single drippy drop of blood. And so what we see happening here is that there is some blood left in the heart, and we call that blood that's left in the heart the end systolic volume, or the ESV, meaning we're at the end of contraction. We're at the end of contraction, and this is the blood that's left over. We're at the end of contraction, and this is the blood that's left over. Okay, so that's the ESV. All right. I've talked for a while, so I want to stop here and stay tuned for the next video. As always, take care of yourselves and each other.